we can manipulate human brains by tampering with the senses, bathing the brain in chemicals, or by playing psychological mind games. But all these methods influence the brain from the outside in. A true mind hack, one that opens us up for reprogramming of any kind, may require something more audacious. Reaching inside a living brain and flicking a switch. MIT engineer, physicist, and neuroscientist Ed Boyden has spent his career figuring out how complex machines work. He's now working on the most complex machine of all, the human brain.
A cubic millimeter of brain tissue has 100,000 cells, and there are a billion connections between them. These cells compute using electricity, and if all of these little electrical computers could be recorded, perturbed, controlled, and analyzed, then maybe we could understand how the brain computed in the same way that computers are engineered to do all the things that they do in our phones and on the internet and so on. So if you have a complex system, whether it's a computer or a city or the economy or a brain, one of the ways that you can figure out what's going on is to actually put little inputs into the system and see what happens.
It's easy to test the components on a computer circuit board, but brain cells are a little trickier. So Ed set out to find a natural switch that can turn individual neurons on or off. And he found one, swimming around in a puddle of pond water.
This kind of algae actually has this light sensor. It sits in a little eye spot in the back of the algae. And basically, these are little light-activated proteins known as opsins that sit in the membranes of cells and serve as photosynthetic or photosensory protein. And this is a molecule that converts light into a very specific change in electrical potential, the exact same kind that occurs in neurons when they're active.
Ed and his colleagues extracted from the algae the genetic code that produces these light-sensitive opsins. They then inserted this gene into a virus that could splice itself into the DNA of living brain cells.
The neurons in the brain will manufacture the protein because now the genome has been incorporated into the nucleus of those cells and then express those proteins all over the membranes or boundaries of those cells.
Ed's virus instructs any infected neurons to build a light-sensitive on-off switch. With these switches, any brain circuit, from our senses to our emotional state and memories, could be controlled with this.
So this is a laser that we developed here, which will then split that laser light out into an array of optical fibers so that we can enter information into the brain into many different points that are distributed in a three-dimensional pattern. The brain doesn't feel pain, so we can implant little probes into the brain, deliver light out the end of these probes, and then we'll turn on or off parts of the brain just by pulsing this laser, turning the laser on and off very rapidly.
Ed and his colleagues first put their technology to trial with animal subjects. Ed wired his light-sensitive switches into a mouse's pleasure-reward brain circuits.
We put in an optical fiber into the brain such that it could deliver light to those cells. Then we programmed a computer such that whenever a mouse poked its nose into a little sensor, it would get a pulse of light.
Each pulse of blue light activates the same pleasure-reward circuits that turn on when the rodent eats a piece of food or finds a mate. The mouse keeps poking its head into the hole, convinced that this simple action is the source of the reward.
And so what we found was that mice would actually work for light. They would poke their nose in the little sensor, get a pulse of light, say, hey, that was interesting, and then do it over and over again, basically doing tasks in order to get a pulse of light. And so what this tells us is that very brief activation of this set of cells with a pulse of light is enough to reinforce whatever the animal was doing just before.
Ed has hacked the mind of a mouse and changed its behavior. And he believes it won't be long before we are ready to use the technique on the human brain.
Arguably, humans have been, hacking the brain for a very long time. Beer, coffee. Those are examples of neurotechnologies that their main role is to change what the brain does. Drugs, pharmaceuticals have been very, very impactful in treating brain disorders. But if you think about the brain as a circuit, then a drug that goes throughout the entire brain is going to affect circuits that you want to leave intact as well as circuits that you want to fix. And so that's where our technologies come into play. We really can make a subset of those cells activatable or silenceable with our tools.
Ed's technology could be a cure for the nearly 1 billion people who suffer from mental illness by turning off only unhealthy cells. It would be a revolution in psychiatric medicine. And one day, it could give us all complete control of the workings of our brains. The rewards for hacking into our brains could be momentous. Mental diseases could be eliminated. We could unlock the highest levels of talent. And we could truly understand what another person feels. But there is a dark side to this technology. We could be creating a future where no one is sure of who they are. No secrets could ever be safe. And where we might all lose our minds to brain hackers.